Okay, so two fundamentally different types of cells. At one time, it was common practice to try to classify all living organisms as either plants or animals. With advances in our knowledge of living things, it has become obvious that the living world is not that simple. Fungi and bacteria, for example, are very different from animals and plants and from each other. Eventually, it was discovered that there are two fundamentally different types of cells. The most obvious difference between these types is that one possesses a nucleus and the other does not. So basically, first of all, let me tell you what is taxonomy. Taxonomy is the study where you classify organisms organisms according to different features, and the person who studied taxonomy is called a taxonomist. So taxonomy, you classify in different ways through either genetic or through nucleus or position of nucleus or not, through different features. So you have a taxonomy chapter later in this book, so then you will understand that in great detail. But right now, just know this, that two different types of organisms are prokaryotic and eukaryotic. Prokaryotes are those organisms that does not contain a true nucleus, while eukaryotic are those organisms that contains a true nucleus. Okay, eukaryotics contains a nucleus, prokaryotic do not contain a nucleus. So what does a true nucleus mean? A true nucleus must contain a nuclear membrane, the DNA, and the nuclear membrane contains nuclear pore, nucleoplasm. So but what does prokaryotic contain? It contains only DNA moving around in the cytoplasm. So you'll learn taxonomy in the later chapters, but for now just understand this much, because this is your first chapter. Nothing will be explained in detail, right? So organisms that lack nuclei are called prokaryotes. They are on average about 1,000 to 10,000 times smaller in volume than cells with nuclei and are much simpler in structure. For example, their DNA lies freely in the cytoplasm. Organisms whose cells possess nuclei are called eukaryotes. Their DNA lies inside the nucleus. Eukaryotes include animals, plants, fungi, and a group containing most of the unicellular eukaryotes known as Prokaryotes. So the Prokaryotes, by what features does Prokaryotes, animals, plants, fungi differ, you will learn in your taxonomy chapter. Most biologists believe that eukaryotes evolved from prokaryotes 1,500 million years after prokaryotes first appeared on Earth. We mainly study animals and plants in this book, but all eukaryotic cells have certain features in common. A generalized prokaryotic cell is shown in figure 1.30. A comparison of prokaryotic and eukaryotic cell is given in table 1.2. Okay, so let's see. Structures always present in a prokaryotic cell are cell wall, cell surface membrane, cytoplasm, circular DNA, and additional structures that sometimes may be present in the prokaryotes along with the structures always present are flagellum, capsule, infolding of cell surface membranes, plasmids, pili. Okay, so plasmid, right? What is a plasmid? Plasmids are simply, look at this. This is circular DNA. That's it. That's what plasmid is. This plasmid is very important in our biotechnology department, like in our biotechnology work. We use this plasmid to make genetically modified organism. We will also learn about that, also how we use plasmid to make insulin. And circular DNA, look, no nucleus, only circular DNAs. Okay, so sometimes referred to as chromosomes, but not always these are chromosomes, and no nuclear membrane, no nucleoplasm, nothing. So this is what prokaryotic cell looks like. And now let us look at the differences. The differences are, as you can see here, prokaryotes and eukaryotes. Prokaryotes, average diameter of cell is 0.5 to 5 micrometer. Eukaryote cells commonly up to 40 micrometer diameter and commonly 1000 to 10,000 times the volume of prokaryotic cell. DNA is circular and lies freely in the cytoplasm. As I have mentioned, DNA is not circular and contained in a nucleus. The nucleus is surrounded by an envelope of two membranes. So yeah, nucleus is a double membrane organelle. We know that. DNA is naked. DNA is associated with proteins forming structures called chromosomes. So the DNA in the eukaryotic cells are actually associated with proteins called histones to form into a structure called chromosomes. But the DNA in prokaryotic cell is naked. It is not associated with proteins or anything. It's just DNA, you know. So yeah, that's a difference. Slightly smaller 70 Svedberg ribosomes, about 20 nanometer diameter than those of prokaryotes. Slightly larger 80 Svedberg ribosomes, about 25 nanometer diameter in than those of prokaryotes. So yeah, so the ribosomes in eukaryotic cells are also slightly larger. This is the unit that we use, Svedberg S. So to measure the bigness or smallness of a ribosome, you don't need to learn this unit, but yeah, you need to know ATS. So I just told you the name so that you know. No endoplasmic reticulum in prokaryotes. Endoplasmic reticulum present to which ribosomes may be attached. So we saw that in prokaryotes, ribosomes are mainly freely. It is not. Remember in eukaryotes, the rough endoplasmic reticulum contains ribosomes attached to it. Also, free ribosomes are present in the cytoplasm, but most of the ribosomes are present in the rough endoplasmic reticulum. But look at this: in prokaryotic cell, there are no endoplasmic reticulum. The ribosomes are just freely moving into the cytoplasm. Okay, very few organelles, no separate membrane bound compartments unless formed by infolding of cell surface membrane. So very few organelles. What does compartment? Remember we learned compartmentalization and division of labor. Compartmentalization is basically forming compartments of different organelles like mitochondria, nucleus, chloroplast. And what is division of labor? Division of labor means dividing the work to different compartments, like chloroplasts. Chloroplasts mainly function as photosynthetic area. Photosynthesis occurs there. Mitochondria, aerobic respiration. Nucleus contains the genetic material. They have division of labor and compartmentalization, but no compartmentalization in prokaryotic cell. Yeah. So, but in eukaryotic cell, many types of cell organelles present. Okay. Extensive compartmentalization and division of labor. Some organelles are bonded by single membrane, some organelles are bonded by two membrane, double membrane, some have no membranes. So yeah, that's complex. Eukaryotic cells are definitely more complex than prokaryotic cells. Eukaryotic cells are more complex and bigger than prokaryotic cells. So yeah, prokaryotic cell wall present. Wall contains murin, a peptidoglycan. A peptidoglycan is a polysaccharide combined with amino acids. We will learn this in the next chapter of macromolecules. Okay, so cell wall sometimes present, like plants contain cell wall, but animals don't contain cell wall. But both are eukaryotic because both possess a true nucleus containing nuclear envelope, nucleoplasm, and the DNA inside the nucleus. The DNA is associated with histone proteins. Yeah. But cell wall is always present in prokaryotic cells. Example in plants and fungi contain cellulose or lignin in plants and chitin in fungi. Okay, that's also a different look. So yeah. Now we will look at viruses. Viruses are different than prokaryotic and eukaryotic cells. In 1852, a Russian scientist discovered that certain diseases could be transmitted by agents that, unlike bacteria, could pass through the finest filter. So it is even smaller than the prokaryotes. This was the first evidence for the existence of viruses, tiny organisms which are much smaller than bacteria and are on the boundary between what we think of as living and non-living. Unlike prokaryotes and eukaryotes, viruses do not have a cell structure at all. In other words, they are not surrounded by a partially permeable membrane containing cytoplasm with ribosome. They are much simpler in structure. Most consist of only a self-replicating molecule of DNA or RNA which acts as a genetic code and a protective coat of protein molecules. See, only this genetic material and the protective coat. Now you may have confusion that then how does it survive? How it does not have any organelles? How does it carry out its function? Yes, you will learn that in I think in immunology chapter. You have learned how pathogens affect, viruses affect. So there you learn that actually when viruses are outside the cell, they are dead. They cannot carry out any of their metabolic process. But when they're inside the cell, this genetic material, if it's a DNA, then the DNA will be used. Oh, you also will learn about transcription and translation, how DNA translates its information to make these specific proteins in the translation and transcription chapter. So you do not know anything yet, but as you go, you will understand. So yeah, this DNA will be transcription and transcription will occur to make the proteins. Those proteins then will do the work for the virus, and also it will replicate to make more of its viruses by using its host's replication machinery. Like the DNA will use the ribosomes and the enzymes of the host cell. It doesn't contain anything, so they are really pathogenic. Okay, so you will learn in great detail in the later chapter, but now this much is enough. Okay, we did that. Now figure 1.1 shows the structure of a simple virus. It has a very symmetrical shape. Its protein coat or capsid is made up of separate protein molecules, each of which is called a capsomer. So the protein coat is called a capsid, which is a polymer of capsomer. Okay, that's a good information. Viruses range in size from 20 to 300 nanometer, about 50 times smaller on average than bacteria. Bacteria are prokaryotic, they are smaller than eukaryotic, and viruses are even smaller than prokaryotic. So yeah, that's you see, first plants and animals, now you see the eukaryotic, prokaryotic, and then these viruses. You will see many classification of animals in your taxonomy course, and that is very important. Taxonomy is actually very important for our biology students, as we need to classify each organism. There are millions and millions of organisms. We cannot just remember all of these structures and everything, so we classify them in groups. Okay, so yeah, you will learn that. So viruses range in size from, I just told that all viruses are parasitic because they can only reproduce by infecting and taking over living cells. The virus DNA or RNA takes over the protein synthesizing machinery of the host cell, which then helps to make new virus particles. Now I said that if it contains DNA, the DNA will be transcribed and transcripted to make the protein. But if it contains RNA, first reverse transcription will occur to form the DNA, and then transcription and transcription will occur. Okay. Also, the RNA can form complementary bonds with the nucleotides to form into a DNA. It depends on the mechanism that the virus is used, but you will learn it in great detail in your immunology chapter. Okay.